Hi everybody, welcome back to another video. So in this video we're going to talk about the regulation and control of breathing, and it'll be a relatively simplified video because this is an extremely complex topic, and so we're not going to really talk too much about the different types of receptors that will affect this and the complex neural regulation side. If you want a more complex video focusing on that, comment below and I'll make that. But this will be a more simplified explanation. Still pretty difficult, but a little bit of more simplified view of things. If you guys like the video, please comment, like, and subscribe. And let's get started. So before we get to talk about any of these things, I want you guys to just realize what we're going to talk about today, okay? So first things first, here's going to be the schematic of what we're going to talk about. So I've gone ahead and just drew this out real quick. Here is the kind of pathway we're going to follow for today, okay? There will be an initial stimulus, okay? That stimulus will trigger the activation of a sensor or a receptor. That receptor will then send, carry that stimulus signal towards an integrating center, meaning that this place is going to sift through the information, integrate the signal, and then it'll pass it on towards some sort of an effector, okay? Then that effector will carry out the response that we need. So let's go through an example of this, okay? Let's say we have a low partial pressure of oxygen, okay? We have a low partial pressure of oxygen. Some sort of receptor will pick that up, okay? Which means it'll increase the activity of that receptor. Then it'll send it towards an integrating center. You guys know that in our body, the integrating center for most of these is going to be actually within the brain, but a specific part of the brain called the brainstem. And an even more specific part of the brainstem called the medulla oblongata, okay, or the medulla. So it'll go to the medulla or the medullary respiratory center, okay? And there it will get integrated, okay, that response, that, okay, we have a low partial pressure of oxygen. Then this respiratory center will go out and say, hey, I need to increase ventilation. I need to increase my inspiration. So what does it do? It'll go to an effector and say, hey, increase your activity. What kind of effector? Maybe inspiratory muscle, for example, the diaphragm. It'll tell the diaphragm to increase its activity and contract more, which means what? We will have a higher inspiration, and the response would be that I bring in more oxygen, which will then, as you can see, inhibit the initial stimulus, okay? So this is the kind of pathway or schematic that we're going to be working with today. All right, and we're going to give a name to all of those stimuli, those receptors, the integrating centers, and more, all right? Just for your understanding, though, many things can actually affect the regulation of respiration, okay? So we have these receptors that are located all over our body. These receptors are practically just sensors. They sense specific things, okay? Specific stimuli. And once they sense that, they will increase their activity. But what does that actually mean? So very briefly, a receptor, okay? Let's say here's a receptor right here. This guy's a receptor. A receptor is very frequently coupled to a nerve, okay? Let's actually draw it like this, okay? Here's a receptor, boom, and this guy is picking up oxygen, the concentration of oxygen. If the concentration of oxygen is low, this receptor will, which is again coupled to a nerve, right? Here's the nerve, boom, boom, will start to increase its activity. If it increases its activity, the nerve that it's coupled to will increase the action potentials down its axon. And what that means is, when it does that, the activity of this nerve is high. And now that the activity of this nerve is high, it is going to carry that information that was picked up by this receptor all right, towards the integrating center, which is the brain, okay? That's what I want you to understand about receptors, okay? They are coupled to nerves, and those nerves will get activated and send action potentials down their axons when the receptor is activated by the stimulus, okay? But what kind of receptors do we have in our body? Well, the kinds that affect respiration are going to be metaboreceptors, they're going to be mechanoreceptors, there are going to be pulmonary receptors, okay? And they're going to be also chemoreceptors, which is the main type that we're going to talk about today, all right? But do realize that we have these three as well, the metabo, the mechano, and the pulmonary, but we're just not going to talk about them in this video, at least too much. Maybe I'll touch a little bit on the pulmonary receptors. And again, if you want a separate video going into more detail on regulation of respiration, let me know. But today we're going to talk about chemoreceptors. And really we have two types of chemoreceptors. We have a central and a peripheral. And the naming is on purpose, okay? The central chemoreceptors are located in your central nervous system, which you guys know is just your brain and spinal cord. And the peripheral chemoreceptors, well, they're not located in the central nervous system. They're out somewhere in the periphery. And as you will see in about a few minutes, they're actually located in the aorta, which is an artery, and another artery called the carotid artery. And you'll realize why they're located there in a second. But those are the main ones that we're going to be focusing on today. We're going to focus on what stimulates the activities of these chemoreceptors, central and peripheral. We're going to look at where they're located, okay? And we're going to look at what kind of neurons are they coupled to, and where do those neurons go in the integrating center, aka the brain, and how does the brain go through that information and cause us to inspire or expire, okay? So that's what we're kind of going to be looking at today. All right. Now, let's take a very quick snapshot and scenario. Let's say we have hypercapnia, which just means we have high CO2 in the blood. Well, if we have hypercapnia, the goal is going to be increasing the ventilation. Okay? Well, why would I do that? Well, if I have high CO2 in the blood, that means that I'm going to have a high amount of CO2 inside of the alveoli. Okay? And I don't want that. So what I do is I expire a lot of it out. Okay? Or my body is going to force me to expire a lot of it out so I can eventually lower the amount of CO2 out in the blood. Okay? That's the main goal here. The blood CO2 is tightly regulated, and I want it to go back to normal levels. So what I do, I increase my ventilation, so I breathe out a whole bunch of CO2. But let's take the opposite scenario. I have hypocapnia, meaning I have low CO2 in the blood. Well, if I have low CO2, my body somehow knows to inhibit, this symbol means inhibit respiration, okay, so that my arteriolar CO2 starts building back up again. Isn't that interesting? How does the body know how to do that? How does it sense the CO2 levels? How does it tell the brain to tell the skeletal muscles of inspiration and expiration to act accordingly? We're going to look at that. We're going to look at that today. In order to understand this, we have to look at a specific part of the brain. So you guys know here's the brain, right? And then here's the cerebellum, and then here's the brainstem, and then we go off onto the spinal cord, okay? So this portion would be the spinal cord, okay? This portion is the cerebellum, this is the brain, and this portion right here is the brainstem, which you guys know is made out of the midbrain, the pons, and the medulla, in that order. After the brainstem, we go to the spinal cord. What I've gone ahead and done in this diagram is I've gone ahead and zoomed in, okay, on the medulla right here. This is the medulla right there. And then as we leave the medulla, we go out into the spinal cord. But you might be wondering why the spinal cord looks like this. Well, in this image, we're taking a look at a cross section of the spinal cord. So imagine I take the spinal cord, I cut it in half right here, and then I rotate it so that you can look at it from a bird's eye view. Okay? That's what it would look like. So that's what we're looking at. All right? And all this blue stuff around this actual medulla, or the brainstem really, is going to be what's called the cerebrospinal fluid. It's basically just this fluid that nourishes the brain, okay? It just bathes the brain, okay? That's all you need to know, all right? And this fluid is going to be pretty important in a second. We'll talk about that. Now, let's go ahead and take a deeper look at some of the specific neurons that sit within this medulla. So let's say right here, this portion is called the medulla, and specifically, for our purposes, it'll be the medullary respiratory center. Okay, now guys, the respiration is really just driven by a whole bunch of neurons and muscles. But how do these muscles contract? They need neurons, okay? But the neurons that tell those muscles to contract need other neurons to tell them, hey, you gotta go and tell that muscle to become active. How does all that work? Okay, well, there's a specific group of neurons within this medulla, or this medullary respiratory center, that we have to talk about. The first one being this green guy, okay, is called the DRG. And then there's another one here called the VRG. And they're both right here, okay? We're just going to write down some notes about them over there. DRG and the VRG. So why are they called the VRG and the DRG? Well, this entire thing stands for the ventral respiratory group. This one stands for the dorsal respiratory group. Okay, if you guys know a bit about your anatomy, you'll know that ventral means anterior or front. Dorsal means posterior or back. Okay, so these names are just saying that both of these guys are involved in respiration, their respiratory groups, and the V and the D are just talking about their location. So the ventral respiratory group is located ventrally or forward. The dorsal respiratory group is located backwards or posteriorly. Cool. Okay, so that's just their names. Now, these guys right here, I said that they're just a group of neurons. But what kind of neurons? Are they inspiratory neurons? Are they expiratory neurons? Well, the DRG is mainly going to be inspiratory neurons, okay? Whereas the VRG has both inspiratory neurons and it has expiratory neurons as well. So that's interesting. Also, the DRG, you guys will see as the rest of this video, not only does it receive a lot of input from many different locations, but it can also go ahead and activate or stimulate, okay? So I'll put plus side for stimulate. It'll stimulate the activity of the ventral respiratory group, okay? So once this guy in and of itself becomes activated, it'll go ahead and activate the VRG as well, all right? So it has the ability to do that. Now, okay. What about the VRG? Any other cool stuff about the VRG? Well, I'm glad you asked, because the VRG, it has another cool fact about it. On top of having this inspiratory neurons and expiratory neurons, it has a specific center, if you will. This is called the pre-Botzinger complex. What does the pre-Botzinger complex do? Okay, well, the pre-Botzinger complex, it is mainly and widely recognized as setting your respiratory rhythm. Okay, sets your respiratory rhythm, your RR. Okay. In other words, it also sets eupnea. Have you guys heard of eupnea? Eupnea is just a term, a fancy word for normal, quiet breathing. Okay, meaning I'm not forcing my inspiration or expiration. And it's about 12 to 16 beats per minute. Now, an interesting thing about these neurons inside of the pre-Botzinger complex is that they have what's known as pacemaker abilities. Pacemaker, meaning that they spontaneously generate a rhythm, okay? And that rhythm, being that 12 to 16 beats per minute, will set your normal respiratory rhythm, okay? Another cool fact about the VRG is that it's not always active. So you know when we breathe, when we're quietly inspiring, okay, so normal quiet inspiration, it's actually the DRG's job. Okay, so the DRG will become activated and it'll inspire, and it'll activate the inspiratory neurons of the DRG, and they will go and do their job. But we're not actually activating the VRG there. And you might be thinking, well, okay, how does expiration happen then? Expiration simply just happens by turning off these inspiratory neurons. And then the lung, as you guys very well remember, will just passively recoil. So you might be thinking, when do we actually use the VRG? Well, the VRG is kind of used as a overdrive mechanism, if you will. Okay? Overdrive. Where when you need more ventilation to support a specific function, you'll touch on the VRG as well as the DRG. What do I mean? What scenario? Let's take exercise. Let's say you're doing heavy exercise, okay? You need to breathe in more and you need to breathe out a bit more forcefully and faster. So what do? As well as turning on the inspiratory neurons of the DRG, you also recruit the VRG inspiratory neurons, so you take in more air collectively using these two. And then something funny is, when the inspiratory neurons of the VRG turn on, they actually inhibit the expiratory neurons of the VRG, and vice versa. So when the expiratory neurons of VRG turn on, they inhibit the inspiratory neurons. That's called reciprocal inhibition. Anyways, during exercise, you need to inspire more. So not only do you recruit the DRG inspiratory neurons, you also recruit the VRG inspiratory neurons. But then as you're about to breathe out, you also have to breathe out more forcefully. So what do? Not only do you turn off the inspiratory neurons to allow for a passive recoil, you also recruit some accessory expiratory muscles. How? By turning on the expiratory neurons of the VRG. Those guys will go out to those muscles we talked about in our previous videos, such as internal intercostals or the abdominal muscles. Okay? And again, we'll take a look at that in a bit. Okay? I'm just giving you an introduction. So that's how the VRG and the DRG are kind of working. Okay? And those are the scenarios that they kind of work in. All right, so a little bit more about those receptors that I was talking about, the central and the peripheral. Well, I said that the central chemoreceptors are actually found in the central nervous system, which we know is the brain and the spinal cord. And we know that the medullary respiratory center is in the brain. So this red guy right here would be called the central chemoreceptors. Okay, cool. Because they're in the CNS. Where would the peripheral chemoreceptors be? I told you that they're found in the periphery, specifically the aorta and the carotid bodies. Okay, well, where's the aorta? Right here. These guys are called your aortic bodies. Okay? And this guy right here, right at the bifurcation point between the internal and external common carotid artery. Okay? Right here, these guys are called your carotid bodies. Your carotid bodies. Together, both of these guys are going to be called your peripheral chemoreceptors. Okay? Together, both of them are called the peripheral chemoreceptors. Something that is very interesting between these two guys is that between the carotid and the aortic bodies, the carotid body seems to be just more active than the aortic body, and we're not entirely sure why. Okay, but it just seems to be more active. Now, another cool thing about these guys is that they have these specialized cells inside of them called glomus cells. Okay, that's how you spell it, glomus cells. And the glomus cells are the ones that are going to actually pick up the change in stimuli. And we'll talk about what that stimuli is in a second, actually. Now, what do the central chemoreceptors and the peripheral chemoreceptors respond to? What is their stimuli? Because remember, we said that it has to be a stimuli first, then the receptor picks it up, okay? And then it sends it to the integrating center. So what is the stimuli that activates the actual receptors? Well, let's take a look. Here's the central chemoreceptors, okay? And then in blue right there is going to be our peripheral chemoreceptors. And we'll look at the stimuli that actually inhibit and activate these guys. And you'll see that it's actually pretty easy to remember. Now, sorry. Let me give me a second. Let me just. So when we're talking about the central chemoreceptors, it's pretty simple actually. There is really one stimulus, but there's a little bit more caveat to it, and I'll talk about it when we talk about the pathway. But really, a high concentration or a high partial pressure of arterial CO2, okay, will stimulate the activity of these chemoreceptors, these central chemoreceptors located in the brain. And guess what? The exact thing that actually stimulates it will inhibit it. So a low amount of partial pressure of arterial CO2 will inhibit it, okay? So a high amount will stimulate it, a low amount will inhibit it. What about the peripheral chemoreceptors? Well, they're located in the periphery, okay? Their stimuli are going to be a little bit different. They're going to be a little bit different. Firstly, of course, a high amount of arterial or CO2 will activate these guys, okay? Another thing, though, that actually happens is that a low partial pressure of arterial oxygen, okay? Low partial pressure of arterial oxygen stimulates these guys. And we'll talk about this. This one's actually very cool and very specific. Another thing, though, is a low pH. Okay, so let me tell you something about pH real quick. When we talk about pH, we are really talking about the concentration of hydrogen ions inside of that blood, okay? And if the concentration of hydrogen ions is high, the pH is low. If the concentration of hydrogen ions is low, the pH is high, okay? So when I say there's a low pH, what am I actually saying, guys? I'm actually saying that there is a high amount of protons, okay? Now, here's the thing. We'll talk about it when we get to the pathway again, but the high amount of protons is actually not due to CO2, okay? You might be thinking, what the hell is this? What are you even talking about, man? But I'll tell you what I mean once we get to it. And the things that inhibit this peripheral chemoreceptor, they're going to be the exact opposite, okay? So for example, a low partial pressure of arterial CO2, a high partial pressure of arterial oxygen, and a high pH, which, as you guys now know, means low protons, okay? Low protons. Awesome. So now, let's take a look at the simpler pathway. Let's look at the central chemoreceptor pathway and figure out how is this stimulation actually causing an increase in ventilation? And what is that even doing? Okay? So let's come down all the way over here. You guys know that everything in your brain, everything in your body receives a blood supply, okay? So here's a blood. Right? Here's a arterial, okay, or a vasculature, whatever, okay? It's in the blood. And we said that we are going to actually have high amounts of CO2 in the blood, okay? That's why we have a high partial pressure of CO2. That was the stimulus, okay? Do you guys know that charged things, such as ions, like, let's say protons, they can't actually penetrate the actual blood here, okay? They can't actually penetrate this space. And that's because they are charged. They have a charge on them. But CO2, it's a lipophilic structure, okay? It can just go through this membrane that's, on this blood vasculature pretty easily. Now, I have some terms written up here. Again, I told you at the start of the video that the blue fluid here is actually the cerebral spinal fluid, okay? And you can think of it as just this fluid that bathes around the actual brain. Then here's the blood, and then there's this thing called BBB. That stands for the blood-brain barrier, okay? Everything that wishes to get into the brain has to cross the blood-brain barrier, and certain things just can't do it, such as this proton. But this CO2 can. So if we have a high amount of CO2 inside of this blood, okay, it can start to move its way inside, okay? And let's actually make this a bit smaller. It'll make its way inside of this cerebrospinal fluid. And then there would be this high amount of CO2 inside, okay? You guys know that the majority of the cerebrospinal fluid is actually just water. So the CO2 that just came in will start to combine with the water that's abundantly present here, okay? And something funny occurs. This CO2 and water, when they combine, they will actually form a substance, or an acid actually, called carbonic acid. They'll form something called carbonic acid, H2CO3, okay? This reaction was catalyzed, or sped up, I could say, by an enzyme called carbonic anhydrase. And if you guys watched the previous video on, hemoglobin transporting oxygen and CO2, you guys will know the reaction I'm talking about. So remember, guys, we had high CO2 in the blood. That high amount of CO2 made its way towards the cerebral spinal fluid, okay? And it made the reaction go this way, in this red arrow. And it made a whole bunch of this carbonic acid, okay? This carbonic acid will just naturally, in and of its own, dissociate. Dissociate into what? Well, it'll dissociate into something called a bicarbonate ion, which is negative charge on it, and this other special molecule here, ion, and that special ion is actually going to be called a proton, okay? So you see how this H2CO3 dissociated into this proton ion. Remember something cool I said in the beginning? This proton ion that was out here, it can't actually make it inside of this brain. So what ends up happening is we actually end up transporting this guy like this, okay? Now, you might be confused about what I just said to you, and that's okay. I'll explain it in a second, but let's carry on the story for one more minute. This high proton that we just created, it's actually going to be a stimuli. It's actually going to be a stimuli for this central chemoreceptor. So look at it. It'll stimulate the central chemoreceptor. All right, let's take a pause here. You guys might be thinking, well, Keon, you said that the central chemoreceptor is stimulated by a high partial pressure of CO2. But you just said that the proton is stimulating it. Isn't that a pH thing? Well, technically, you're right. It could be a pH thing, okay? There is a pH change at the level of the cerebrospinal fluid. But really, what made this reaction go in this direction was because of the high partial pressure of CO2 in the blood. The partial pressure of CO2 was so high that it made this CO2 come out into the CSF and drive this reaction that way. Why did it make the reaction go this way? Because this reaction is called an equilibrium reaction. Meaning what? Meaning the reaction is reversible based on the amount of substrate that you have on either side. So if I have a lot of bicarbonate and protons, I can drive this reaction this way. If I have a lot of CO2, I can drive this reaction that way, which is exactly what I just did. Okay, so now you guys know why this reaction had to go this way, all right? It had to go that way. All right, so we still can say that it's due to the partial pressure of arterial CO2, but sometimes you might see that a high pH is also, a low pH is also a stimulator, okay? Sometimes you might see that, but just keep in mind that it's actually talking about that proton concentration, which is actually just due to the CO2. That's why we just kind of disregard it, okay? All right, cool. Now, what happens when this central chemoreceptor gets stimulated? Because again, remember, we had our stimuli, then we had the receptor picking it up, then we actually had the integration center. Okay, we had the integration center, then we had the effector, and then we had the response. So let's go through that. The stimuli was the high CO2. The receptor was the central chemoreceptor. What's the integrating center? The integrating center is going to be the DRG. So look at this. The DRG will get stimulated, right? The DRG will get stimulated. And then the DRG has these neurons within it. If you guys remember from the start of the video, I said that the DRG has these inspiratory neurons in it. These inspiratory neurons will go all the way down to the level of the spinal cord. they will synapse on these other neurons in the spinal cord. These other neurons in the spinal cord, they're called motor neurons. They're very special. They come out to your muscles, your skeletal muscles. What kind of skeletal muscles? In this case, it's going to be your diaphragm. You guys know what your diaphragm is responsible for? It is an inspiratory muscle. Okay, so look at what happened. The DRG, having inspiratory neurons, increases after being stimulated by the central chemoreceptors. It increases these action potentials down its neurons and activates or stimulates these actual motor neurons, okay? And this motor neuron will then increases action potentials, which will then stimulate the contraction of the diaphragm. If the diaphragm contracts, guys, what happens? You're actually going to inspire more, okay? Or increase your ventilation, I should say. You're actually increasing your ventilation. So the stimuli was high partial pressure of CO2. The receptor was the CCR. The integration was the DRG. The effector was the actual diaphragm. Okay? And the response is increasing the ventilation. How does that alleviate the initial response? Well, if you increase ventilation, guys, remember our example here? Hypercapnia, high CO2. You increase ventilation, what happens? You're breathing out a whole bunch of CO2. If you breathe out a whole bunch of CO2, guess what? Your arterial CO2 will come back to normal, which will eventually alleviate that initial response and turn off this pathway, this negative feedback pathway. That's pretty darn cool. Okay? But you might be thinking, hey, Keon, didn't you say the VRG is also activated when the DRG is activated? It can be, okay? It can be. So, and I told you that during normal quiet breathing, the DRG is just doing its own thing, okay? But we're not talking about normal quiet breathing. We're talking about regulating the respiration. So in what scenarios would your partial pressure of arterial CO2 rise? For example, during exercise. So, and if you remember, we said that the VRG is involved during exercise. So we can actually say that in this scenario, the DRG is stimulating the VRG as well. But what kind of neurons in the VRG? The inspiratory neurons of the VRG, okay? It'll turn on the inspiratory neurons of the VRG. So let's draw that. So here's the inspiratory neurons of the VRG. They're coming down, and they're going to go and synapse on these neurons. What are these neurons? Again, a couple of motor neurons that will go and act on specific muscles. What kind of muscles? Accessory inspiration muscles, like the external intercostals. Okay? The external intercostals. Okay, cool. And then you guys know that when this inspiration is done and expiration is initiated, what happens? The DRG is no longer being stimulated, okay? Why? Why is it no longer being stimulated? Because remember, we increased our ventilation, we lowered our partial pressure of arterial CO2, which means there's no longer a stimuli to the receptor, which means this receptor is not getting stimulated, which means it's not stimulating the DRG, which means the DRG is no longer stimulating these motor neurons, and it's no longer stimulating the VRG. Okay? Well, if the DRG is no longer stimulating these guys, guess what? These muscles are relaxing, and that's going to initiate the passive recoil of the lung. Cool. What else? If the DRG is no longer stimulating the inspiratory neurons of the VRG, what's going to happen? Remember I said there's a reciprocal inhibition? So when the inspiratory neurons of the VRG are stimulated, that inhibits the expiratory neurons. But when they're no longer being stimulated, guess what happens? The expiratory neurons become more active, and they inhibit the inspiratory neurons from working. But now look at this. Let's draw it like this. The expiratory neurons of the VRG come in, and guess what? They're going to come and act on these other motor neurons. These other motor neurons, they're going to go to specific muscles of expiration. Like what? Like the internal intercostals or the abdominal muscles, okay? All right, and that's how it's kind of working during that, what's it called? That exercise scenario that we were talking about, okay? But remember, during regular quiet breathing, it's just the DRG doing its thing. I cannot stress that enough, okay? Alrighty, guys. So, see, that wasn't too bad. That wasn't too bad. Now that you understand this, you're going to have an easy time understanding the peripheral chemoreceptors. The peripheral chemoreceptors, I'm only going to go through one of them. Okay, I'm only going to go through one of them. Well, actually, two of them. The oxygen and the pH. Okay, but not the CO2, because the CO2 is the exact same process as this guy right here. Okay, it's just, instead of happening in the brain, it's actually happening in the blood. Okay, the same reaction. And that's how you're sensing that CO2. But anyways, with the pH, the reason why I'm saying at the start that it's not due to the CO2 changes was because of this reaction. You guys remember how this high amount of CO2 generated these protons, which changes the pH? Well, the pH that this guy is sensing, this peripheral chemoreceptor that it's sensing, it's not due to the CO2, because that's written over here. The CO2 is right there. That's how we're sensing that proton change. But this proton, this pH change, is not due to the CO2. So what is it due to? You guys know when we exercise? When we exercise, we actually make a specific molecule called lactic acid. Okay? Lactic acid, though, what a lot of people don't know is that it's actually written like this, HLA. It's made up of two things. It's made up of this hydrogen and this lactate ion. During exercise, you make this lactic acid, and this lactic acid makes its way into the blood, and it separates. It separates into two parts, a lactate ion and a hydrogen ion. Forget about this lactic ion, okay? You don't have to worry about what this does. This actually contributes nothing to fatigue. This guy, though, this guy contributes to fatigue, and also this right here is one of the changes in pH that these peripheral chemoreceptors would be sensing, okay? So when I say it's not due to CO2, it's due to other acids, one of them being lactic acid, okay? Because look, it actually generates those protons. Okay, so now you know what I mean when I say not due to the CO2. Okay, so that's that change with the protons, okay? And the response is going to be pretty much identical with the oxygen that we're going to talk about. It's just that the oxygen has a little bit of a caveat to it. So far, we've just been talking about arbitrarily high amounts of this and low amounts of that, okay? And it's just because the response is so varied, okay? It's kind of like a gradient, all right? But with oxygen, it's very, very, very much so specific, okay? When we say low partial pressure of oxygen, it has to be less than 60 millimeters of mercury. It has to be. And you're thinking, why? Why does this peripheral chemoreceptor have to be so sensitive to a drop in partial pressure of oxygen less than 60 millimeters of mercury, but not anything above it? Why? Let's take a look at the oxygen hemoglobin dissociation curve again. If you remember from that video, I mentioned this. Here's the partial pressure of oxygen in the blood, right? Here's 100, 90, 80, 70, and lo and behold, 60. At 60 millimeters of mercury, take a look at where we're at, and let's change this to black. Take a look at the oxygen, the hemoglobin saturation with oxygen. And let's take a look at 100 as well, while we're at it. What do you notice, guys? What do you notice? Does the percentage of hemoglobin saturation drop drastically between 100 millimeters of mercury of oxygen in the blood and 60 millimeters of mercury? I'm going to go ahead and say no. So over here, we're about 98% saturated, whereas over here, we're 90% saturated. An 8% drop, that's nothing. That's literally nothing, But what do you notice? This portion, if you guys remember, I said this is the plateau portion of the curve. And then the rest of it is kind of like the steep portion. Well, guess what? As soon as you drop below the 60, guess what's happening? You start entering the steep portion. And let's just take 50 millimeters of mercury. Okay? 50 millimeters of mercury, guys. You're no longer saturated 90%. You're saturated 81%. Okay? 81%. Look at the difference that exists between 81 and 98%. That's huge. So the reason why the body is so sensitive, more specifically why those peripheral chemoreceptors are so sensitive to the stimulus of less than 60, is because anything at 60 and higher, we're at this plateau portion of the curve. Our oxygen hemoglobin saturation is pretty good. We don't have to worry about that. But as soon as we get less than this, it's kind of like a protective mechanism or a panic signal. It's like, whoa, we're getting very low. We're getting very low on this oxygen hemoglobin saturation. I need to increase my ventilation to bring in a whole bunch of oxygen to increase the partial pressure of oxygen in the blood back up to greater than 60. Okay? And let's take a look, now that we understand that, let's take a look at the effect of a low partial pressure of oxygen on the peripheral chemoreceptor firing frequency. What do I mean when I say firing frequency? I'm talking about the action potential firing frequency. Because if you remember, I said that the receptors, every single receptor is coupled to a neuron, and that neuron will then increase its action potential frequency. Go back to the start of the video to remember that. So let's take a look at these three scenarios here, okay? So here we have in 1 second, we have in 1 second, and we have in 1 second here, okay? And then we have three different partial pressures of oxygen. We have a partial pressure of oxygen at 40 millimeters of mercury. We have 100 millimeters of mercury. And we have 120 millimeters of mercury. Okay, so at 100 millimeters of mercury, what is the firing frequency going to look like? 100 millimeters of mercury. Well, this is where we're kind of normally at, right? You guys remember 100 millimeters of mercury? Right here. What does it look like there? What is the peripheral chemoreceptor, these guys, right? So this guy and that guy, what does their firing frequency look like down their nerve? Well, it looks something like this. Boom, 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 boom. These guys right here, these are just action potentials, the frequency, the number. 1, 2, 3, 4. Okay, cool. That's their normal rate. What if the partial pressure of oxygen rises up to 120? Okay, so more than physiological levels. It's hella high. Well, I told you that one of their stimulus was low partial pressure of O2. And we said that the inhibitor was a high partial pressure of O2. Okay? So what this means is that their action potential frequency is going to be very, very low. It's very sparse. And again, I'll write this down. Action potential frequency. Okay, cool. Now, what happens though if we drop below that 60 millimeters of mercury threshold? We go to 40 millimeters of mercury. What happens? Take a look at the action potential frequency then. It's going to look like this. Boom, 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 boom. Look at that. I can't even count that. That's how many that is. There is a lot of action potentials going on here, and that's because the body is saying, hey, you are way below what you're supposed to be at. You need to increase right now. Okay, you need to up your partial pressure of oxygen right now. And how does it do that? By breathing in more. And we'll take a look at that right now. But this is a very cool example of demonstrating how sensitive these guys are to that threshold we talked about. So let's zoom in on both of these guys. Here's the carotid and here's the aortic bodies. They are both peripheral chemoreceptors. They're both responding to the same thing. And remember at the start, I said that the carotid bodies are just more active than the aortic bodies, and we just don't know why. Okay? So the carotid bodies, they are coupled to a nerve, and that nerve is going to send its signals down to the DRG, and so will the actual aortic bodies. Okay? Both of them, they're coupled to a nerve. Different nerve. If you're curious, This one is coupled to cranial nerve 10. This one is coupled to cranial nerve 9. Okay, or actually, sorry about that. This one is coupled to cranial nerve 9, which is the glossopharyngeal nerve. Okay, and this one is coupled to cranial nerve 10, which is the vagus nerve. Okay. Now, why? Like, what does this pathway look like? So the carotid bodies, let's say they're sensing, and let's write it like this: they're sensing a low partial pressure of oxygen, less than 60 millimeters of mercury. And same thing for the aortic bodies. They are going to increase at that moment. They're going to increase their action potential frequency. Okay, and you'll see that right here. Increase their action potential frequency all the way down towards the level of the DRG. At the DRG, guess what they're going to do? They're going to stimulate the DRG. Why? Because the DRG has inspiratory neurons, inspiratory capacity. If we're low on oxygen in the blood, what do we want to do? We want to breathe in oxygen, inspire oxygen to up those levels in the blood. So guess what? We're going to stimulate the DRG. The DRG, in turn, will do what? Increase its action potential down here, which will then what? Also stimulate the VRG. The VRG what? Inspiratory neurons. And then look what happens. We're going to stimulate the activity of those inspiratory motor neurons, these two, and they're going to increase our ventilation, okay? Which will do what? Which will increase the oxygen and alleviate this initial stimulus, okay? Inhibit this initial stimulus, all right? That's what that's doing. Now, let's say I am increasing my partial pressure of oxygen to around that 120. Well, guess what? There's going to be fewer action potentials, okay? Fewer action potentials down these guys. And if there's fewer action potentials down these guys, there's actually going to be fewer action potentials all throughout this pathway. Which means what? Which means there's going to be less stimulation of these neurons, okay? Of these neurons. Okay, I hope all that is clear. It's just a very simple pathway that we're kind of following, all right? Now, we mentioned that both of these guys, the central chemoreceptors and the peripheral chemoreceptors, they're both responsible for sensing CO2. But which one senses CO2 more? Well, the central chemoreceptors are responsible for sensing about 70% of that, and the peripheral chemoreceptors are responsible about sensing 30%, okay? So really, you can say that the peripheral chemoreceptors are more of an oxygen sensor, and a central chemoreceptors are more of a CO2 sensor, okay? Obviously, you guys know otherwise, but you can say that to simplify it more for yourself, okay? Now, and again, you guys know, as before, that once we finish the inspiration process that occurred with those PCR response, we will then inhibit the DRG again, right, to initiate passive expiration, and then inhibiting the DRG will inhibit the inspiratory neurons of the VRG, which will stimulate the expiratory neurons of the VRG, which will then go out and stimulate these kinds of motor neurons, which will go out and activate the accessory expiratory muscles. Cool. Now, one last thing that I want to mention is that in certain areas of the lung, okay, such as some of the bronchial smooth muscle, okay, so here's some bronchial smooth muscle, okay, you will find very, very specific receptors called pulmonary receptors. And these pulmonary receptors, they're attached to a nerve. But really, they have a specific name. They're actually called pulmonary slow adapting stretch receptors. Man, that's a hell of a name. But what does it mean? It just means that they sense stretch. What kind of stretch? Well, you guys remember from the volumes and capacities video, the tidal volume? If the tidal volume is greater than 1 liter, and this can differ, okay? But if the tidal volume is approximately greater than 1 liter, that's going to stimulate these pulmonary slow adapting stretch receptors. And they will then send a signal down the nerve that they are coupled to, all the way to the actual respiratory center in the medulla. Think about this. If the tidal volume is greater than 1 liter, what does that tell you? Is the lung overinflated or underinflated? Well, that tells me that it's overinflated, because remember, the regular tidal volume is about 500 milliliters, right? So this is kind of a protective response, if you will. It senses that the lung is actually overinflating, and what does it do? It sends a signal down this nerve. What kind of nerve? This is actually the cranial nerve 10, the vagus nerve, same as this one. It's an afferent nerve. And it goes all the way to the DRG, and it actually inhibits the DRG. It says, hey, you know what, DRG? We don't need to inspire anymore. Okay, we don't need to inspire anymore, because guess what? The lung is overinflated. I'm going to inhibit you so that you can't actually send action potentials down to these motor neurons to activate the inspiratory muscles, which will cause an expiration. And if we're exercising, it'll actually inhibit the VRG signal as well, okay, and activate the expiratory neurons of the VRG. So that's what this kind of reflex is doing. This reflex has a specific name. It's called the Hering-Breuer reflex. And it's a classic example of negative feedback loops. It was discovered around 1860s by this guy named Hering and Breuer. And all it is just a protective mechanism from the overinflation of the lung. Okay? That's literally all it's doing. Now, believe me, you guys, we've went through a lot of stuff here, okay? We've covered a lot of complicated topics, but believe me, it can get even more complicated than this. If you want a video on that, let me know down below, and I'll cover it in full depth, okay? There's way more to this stuff. There's other receptors, there's other neurons within the pons and all that stuff that can actually help with inspiration and expiration. But if you've stuck around this far, I appreciate you a lot. If you gained something from this video, if you truly learned something, please like, subscribe, and comment down below. Again, I cover a lot of things, so this would be a useful channel to have around. And without anything else, I'll see you guys in the next video.